So we continue our today's last session because our research target is how to turn our research to the commercial product. So this session is industry talks, sensor commercialization, cases start. The first talk is Professor Jun Qian from Zhejiang University. His talk is deep tissue and high resolution in vivo microscopy based on near-infrared radiation to tissue window. Please welcome Professor Qin Jun.
Good evening, everyone. First, I'd like to thank Professor Xiong for the kind invitation. And today, the title of my presentation is Deep Tissue and High Resolution in Vivo Microscopy based on NIR-II tissue window. Okay, actually, it is not so industrial talk. It is actually a little bit academic. Okay, but still I will have some industrial component inside my presentation. Okay, so I'm Jun Qian. I'm from Zhejiang University, College of Optical Science and Engineering. Okay, so in our group, we mainly focus on the deep tissue in vivo optical bioimaging technology. So why optical? Because we know optical have lots of advantages. For example, high spatial resolution, high specificity, and free of radiation. And our aim is to achieve deep tissue visualization because this technology can be used for the brain science study and brain disease diagnosis, organ visualization, and imaging guided treatment, and so on. So as we know, biological tissues have the optical absorption and scattering. Both of these two factors will limit the imaging depth and spatial resolution. So for the tissue absorption, it will decrease the intensity of light. So it is called attenuation. So previously, people mainly focus on their interest in optical spectral range, which is from, for example, 760 nanometer to 900 nanometer, because in this range, the tissue absorption is very small, so they can call this window as optical tissue window. Actually, as we know that compared with tissue absorption, tissue scattering impact more on the imaging performance, because besides attenuation, the tissue scattering will also produce the diffusion of the light. Okay, so recently, people transfer their interest to another optical spectral range, which is from 1000 to 1700 nanometer, because in this window, as the wavelength of the light increases, the scattering also decreases accordingly. And in that case, the tissue, we can have less attenuation and diffusion. So this window is called a NIR-II window. Okay, so NIR-II fluorescence in vivo imaging, bioimaging, according to this definition, we know that the wavelength of signal is in the NIR-II range, which is from 1000 to 1700 nanometer. Okay, so it is quite different from the visible fluorescence in vivo bioimaging and also the NIR-I fluorescence in vivo bioimaging. So this concept was first proposed by Professor Hongjie Dai in Stanford University in year 2009. So in this Nature Medicine paper in 2012, Professor Dai's group successfully demonstrated that NIR-II fluorescence imaging have better performance compared to the NIR-I fluorescence imaging. From the you can see both the spatial resolution and the signal to background ratio, and also the depth. NIR-II fluorescence imaging outperforms the NIR-I fluorescence by imaging. And also in this 2015 years work, they defined 1500 nanometer to 1700 nanometer as a NIR-II-B window. And the NIR-II-B fluorescence has the best performance according to their experiment. And also there are lots of other groups doing very excellent study, excellent results in the NIR-II fluorescence area. Okay, so here I just list some. And as we know, there are many research groups in different countries get involved in this study. And in 2019, the first NIR-II fluorescence imaging was successfully held in Xi'an. Okay, so why the NIR-II fluorescence has the advantages? Why it can perform better than NIR-I or the visible fluorescence imaging? Okay, so during the past few years, the mainstream viewpoint is that first, the fluorescence signal has a longer wavelength, so the light scattering is small in the biological samples. In that case, it can help us to improve the imaging depth and spatial resolution. And also the second is that, as we know, the biological tissues has very low autofluorescence in longer wavelengths, for example, in this NIR-II region. Okay, so it can help us to improve the signal to background ratio. However, as according to our work in 2020, here we can see that fluorescence beyond 1500 nanometer performs much better than the fluorescence imaging beyond 1200 nanometer. Okay, but as we can see, the scattering around 1200 nanometer and 1500 nanometer is not so big difference. They are very similar. Okay, so it is a very interesting but also very strange result. Okay, and also we can see this spectrum. Usually people defined NIR-II as 1000 to 1700 nanometer and defined 1300 nanometer to 1400 nanometer as NIR-II-A. Okay, here, and 1500 nanometer to 1700 nanometer as NIR-II-B, like this. Okay, so how about this one? 1400 nanometer to 1500 nanometer. Why didn't they use this range? So according to their viewpoint, because the water absorption in this window or in this spectral range is very large, so they think this spectral range is useless. So is it true? Okay, so then based on this puzzle, we raised two scientific questions. First, whether the existing mechanisms for NIR-II fluorescence in vivo bioimaging are complete. And second, whether the 1400 to 1500 nanometer spectral range can be fully utilized. Okay, so this is ACS Photonics paper in 2016. Okay, so according to their results, they proposed that absorption can increase imaging contrast of biological tissue. Okay, here, the moderate absorption can play a positive role. Okay, and then in our group, we did lots of simulation work like this and also like this, and also conducted experimental work. For the time being, I will not talk about too much about the simulation and experiment details. Okay, and finally, our conclusion is that the tissue absorption can suppress the background caused by scattered light during the in vivo bioimaging. Okay, in vivo or in the tissue bioimaging. Okay, so that's the most important reason why the NIR-II fluorescence in vivo bioimaging can have better performance compared to the traditional fluorescence bioimaging. Okay, this is the first conclusion. And the second, of course, this window, 1400 nanometer to 1500 nanometer, can be fully utilized because here, although the water absorption is very huge, but it can also help us to suppress the background to a certain extent. And in our work, we also extended and perfected the NIR window. Okay, in our work, we defined NIR-II window as 900 to 1550 nanometer. Okay, because here we defined 1700 nanometer to 1880 nanometer as near IRC. It is also a very good window. And for this one, we defined 1400 nanometer to 1500 nanometer as near NIR-II. So YX, okay, because it is determined by the brightness of the fluorescent probes. If the brightness of the fluorescent probes is large enough, so this one is useful. If the brightness of the fluorescent probes is not so large, it's very small, so maybe this window is useless. Okay, so this one is dependent on the brightness of the fluorescent probes. Okay, and also we defined 2080 to 2340 nanometer as the near R3 window. According to our study, this is a very potential window for high performance fluorescence bioimaging. Okay, so then we combined our near NIR-II window with the microscopy. Okay, so here I just listed two examples. First, we can combine our near NIR-II fluorescence imaging technology with the traditional wide field fluorescence microscope. So as we know, the wide field fluorescence microscope based on the area excitation and area detection. Okay, so the fluorescence is in the visible range. Okay, so for the visible fluorescence microscope, they can only be used for the observation of cells and very thick tissues. Okay, however, if we introduce near NIR-II window to this microscope, so we can get deeper penetration, higher spatial resolution, and also we can achieve real-time observation with tunable magnification. Okay. So here are some examples. First, we used fluorescent probes called AIE dots. So maybe tomorrow morning, tomorrow afternoon, Professor Benjong Tang will give some details introduction about the AI dots in his plenary talk. Okay, so here we also collaborated with Professor Benjong Tang and used the near IR AI dots. And we intravenously injected these dots into the body of mouse. And then we did some surgery on the brain of the mouse and put this brain under our microscope. We can observe the blood vessels at different depths. Okay, so depths can reach around 800 micrometer. And also we can use AI dots for the observation of tumor, microscopic observation of the tumor. We can find the difference between the older tumor and the newly grown tumor. For example, in this newly grown tumor, most of the AI dots not only distributed in the blood vessels, and also they can distribute in the surrounding tissues, surrounding tumor tissues. Okay, but for the old tumor, almost all the AI dots are located in the blood vessels, so they are quite different. And also, we can use our optical method to establish a thrombotic ischemia mode inside the blood vessels. So according to the area here, this is a thrombotic ischemia mode. And then accompanied by the blood vessel flowing. Okay, so this ischemia mode will be flushed by the blood flow. Okay. And we also studied lots of AI dots and find one which can be cleared completely from the body of the mouse. And then we can use this one for the intravenous injection to the small monkey called a marmoset. Okay, and then we also conducted the brain blood vessel observation and also measure the velocity of the brain blood vessel. And also we establish a thrombotic mode and successfully do the visualization. And here is another example. Here we used a fluorescent probe named quantum dots. It can give out very bright fluorescence in this near NIR-II window. Okay, and then we did the wide field near NIR-II microscopic imaging. And here we can see the imaging depth can reach around 1.3 millimeter. Okay, this depth is very large, comparable to the ones in the 2-photo fluorescence or even 3-photo fluorescence microscopy. And here is another example. Here we also intravenously injected the quantum dots into the body of a mouse. And then we do the observation of the intestine of the mouse. So the whole one is the intestine of the mouse, and it is wriggling. And also the blood vessels in this intestine can be also visualized very clearly. Here we can see the flowing of the blood. Okay. And then we use our method to do the observation of the distribution of ICG and quantum dots in the liver of mice. Okay, here it is also in vivo bioimaging. We did not take the liver out from the body of the mouse. The ICG, as we know, it is a clinically approved NIR dye. Okay, so it can be cleared from the liver of the mouse. And under our microscope, we can find that most of the ICG are distributed in the cells of the livers, but not the blood vessels. Okay, but for the quantum dots, as we know, and quantum dots are very difficult to be cleared from the body. So most of the quantum dots are distributed in the blood vessels rather than the cells. So maybe that's why the quantum dots are so difficult to be cleared from the body. Okay, so then we collaborated with Sunny Optical Company, and which is a very famous company, and ranked #1 or #2 for the microscope manufacturing. Okay, and we realized the industrialization of our NIR-II fluorescence microscope. Okay, so the second technology is the NIR-II fluorescence confocal microscope. And for the confocal microscope, I think most of you are very familiar. For the confocal, it is based on the point excitation and point detection mode. So compared with the wide field microscope, it has higher spatial resolution, but it also sacrifices the temporal resolution. So for the traditional confocal microscope, its fluorescence emission also locates in the visible or maybe near NIR-I window. Okay, so in that case, they could not penetrate so deep, so only can be used for the observation of cells and tissues, thin tissues. And then if we combine NIR-II fluorescence with the confocal microscope, we can get a new technology called NIR-II fluorescence confocal microscope. It can help us get deeper penetration and higher spatial resolution, and also higher signal to background ratio. So it can be used for the in vivo observation. So here, NIR-I excitation light was used because it can have larger penetration than the visible excitation. And also the NIR-II fluorescence was collected because it can provide higher imaging contrast in tissues. So here it is also AI dots. It was kindly provided by Professor Bingliu's group in NUS. So under the NIR-I excitation, this AI dots can give out a very bright NIR-II fluorescence. And then we intravenously injected these nanoparticles into the body of a mouse.
Qian, only
Yes?
1 minute.
Okay. And then we did the observation of the blood vessel at different depths, and based on this imaging, we can do the reconstruction and get the 3-dimensional architecture of the blood vessels. So here is the dynamic imaging showing the blood vessels at different depths. Here the imaging depth was around 800 micrometer. So we also did some modification of our confocal microscope. We can put it on a shelf with the direction adjustable shelf. Okay, and then in that case, this confocal microscope can be used for the observation of large animal, for example, the big monkey. And here the ICG was used as the probe because it is safe for human being, and it should also be safe for the monkey. Here is the imaging result. So we can observe the brain blood vessels under the confocal microscope and also get the three-dimensional architecture of the brain blood vessel of the monkey. So the imaging depth was around 500 micrometer. And also we can improve our system and set up NIR-II lifetime microscopic systems. And also we did some NIR-II fluorescence lifetime imaging of the brain blood vessels. And here the color indicates the lifetime, which was around 500 picosecond. So here is the as the clearance of the probes, the lifetime also changes gradually. Maybe we can use this phenomena to monitoring this clearance of probe in the near future. Okay, so finally, it is the conclusion. We extended and perfecting the NIR imaging window, and we use lots of fluorescent probes and established optical instruments in our lab, for example, wide field microscope, confocal microscope, and so on. And the different model animals was used, and we did lots of biomedical applications. Okay, this is acknowledgement. For the time being, I will skip this slide. So thank you so much.
Oh, excellent talk. We have one question. If any question. So, Professor Qian,
yeah,
you use NIR-II laser source. Do you use the solid laser, for example, or semiconductor lasers?
Oh, here we use the semiconductor lasers.
How much power normally you need?
Okay, two different cases. For example, for the wide field imaging microscope, the power of the laser is not so large. Maybe, for example, 40 or 50 microwatt before the objective. Okay, this is for the wide field imaging. And for the confocal, so it is another story because we use point scanning. So the laser power is a little bit large, maybe around, according to my knowledge, maybe 60 or 50 milliwatt before the microscope. Yeah, because as we know, the NIR-II fluorescence is not so so bright, so we need to increase laser power a little bit. Yeah.
Thank you very much. So, any question?
Hi, I'm Hu Bingjia from UCL. It's not a question, it's just a quick comment. It seems like I think like your technology is really impressive. I think that definitely have the feasibility to practically apply it for functional brain imaging, which I'm very interested. I just like say like if you are interested, probably we can have a discussion offline to talk about like a possible collaborations if you are interested. Yeah.
Yes.
That's it. Excellent work. Thank you.
Thank you.